Thank you, Mike. I'd like to ask Brad and Susan and Jonathan if they wouldn't mind to come back up, and we have some time built in now to have a little interactive panel discussion and questions. So if pull that out, and we have the microphones here. I'll sit over here. Thank you. And we'll just open it up, but maybe I could just start to throw the ball out there a little bit. So we've heard about molecular biology, molecular pharmacology of signaling pathways and regulating transmitters. We heard about the genetics, underlying genetics of a major neuropsychiatric disorder, and some very promising work. And then we heard from the industry perspective about, frankly, some challenges of really, we haven't got there yet, particularly for these big questions. And so what I'd like to hopefully have here is a little bit of discussion from the people working on the research side and the people on the pharmaceutical industry side about how we're going to converge on these big questions. So for example, on depression, how are we going to be able to take the kind of data coming out of Jonathan's work, or in the cases of effects of drugs and neurotransmitter pathways, how are we going to bring that together? How do you guys in the pharmaceutical industry see that work being supported and coming in and really making a difference? You talked about consortia a little bit earlier, I think, Brad, and I know AstraZeneca has done those kind of things, Mike, when you were with them. So where's the connection going to happen, and how are we going to actually get there? What are your thoughts? So,
perhaps I can just start. So
please, go ahead. Slide the mic close.
Yeah, so I...
Is that on?
Okay, yeah. So I'd really like the discussion you put forward about the digital apps, and we've been thinking on very much the same lines. So I'm part of a large study we're setting up in UCLA called the Depression Grand Challenge, where we're going to recruit about 100,000 subjects, and we're going to be deploying exactly that sort of technology. And I have been a little suspicious about how useful it was until I got some data from our own work, the study we had in China. So what we did there was we recorded 80% of the interviews. This was for quality checking. And there was a computer scientist in Beijing who took these data, and he's been analyzing the speech. So in the same way that you are getting 50 or 60 bits of information from tapping on the phone, he's getting about a 90 or so from each of the words that he's able to recognize that the subjects say. Now, this was not on perfect recordings. These were recordings not made for this reason, so he had to throw away a lot of the data, but still, to my astonishment, he was able to take a question, please tell me about how you were behaving on this particular day. It was slightly emotionally loaded, but not particularly, and just by taking the tone, so nothing to do with the meaning, just by taking the tone of those words, he was able to predict with 100% accuracy whether the patient would later go on to say that they were thinking of killing themselves. I found that a really striking observation, that you were getting that sort of data, the digital data that you're talking about, to put it into something that was really clinically useful. And clearly, if you can do that on a larger scale and get other stuff, I think the approaches that you're describing will be extremely useful. And we'd be very keen on the sort of collaborations that you were mentioning as well. I think it's not possible to do this unless we look very broadly at this whole area.
Thanks, that's exactly. Hello. I think that's exactly what has to happen. If pharma's not very good at starting. Oh, and by the way, Mike, to your comments, the first schedule I had seen did have you going before I did, so we perfectly planned out to swap. Pharma succeeds best when academics does what it does best, which is take their understanding of disease and then develop in a direction that pharma's reaching as well. And the challenge is for everyone to grab hold while pharma's driving by the town. The problems are basically issues of timing. Pharma needs to be able to declare that there's an area of interest, and this goes against their DNA because they try to keep things under wraps for patent protection purposes, at least for individual diagnostics. But pharma's coming around to the understanding that at least in a category of disease, for example, major depression, pharma needs to be able, all interested members to give you enough funding, need to be able to declare that we're interested in further developments in this field that we can then capitalize on internally. Pharma needs to be able to meet with you in a way that's timely for you and for them so that you can do the work that you're doing. And they need to be able to say, you know, here are upkeep areas for us, so digital biomarkers. We've seen the same thing. I haven't talked about voice. We've seen the description of what you've said, also in areas of frontotemporal dementia, aphasias, things like that. A lot of this syntax parsing and voice tone stuff really does seem to be kind of very predictive. So we need to be able to show you the routes that are most productive and then allow academic and government consortia to then fill in that area with knowledge that's actionable. And where everyone needs to get better is on issues of timing, because the problem is pharma will move on, and academics who have worked with pharma have probably all experienced this. Maybe a year or two, things get going, they're very exciting, and then either the pharma discontinues that area of investigation, or there are a couple, to your point, a couple failed phase 2 trials, and all of a sudden interest cools and they won't answer the phone or the email anymore. They may not even be there anymore. So, pharma cycles through reorganizations every 2 years, and very often people in pharma informally reorganize every 2 years. So it's a tough place for an academic who builds a career in one place, developing a single line of knowledge, often in multiple different directions over a career, and influencing the development of generations of scientists and potentially scientist physicians. We're a difficult dance partner, and that timing, I think, has still not been solved. Consortia, the problem is, there'll be an annual meeting, and there'll be a set of work groups, and they'll meet on their own, and then the thing becomes discoordinated. And the problem is pharma won't signal reliably its timelines. They're not going to say, well, we're going to read this out and we expect it to fail and then we're going to go away. Until that timing's solved, the problem is that you'll have two increasingly exuberant partners and something of a discoordinated dance. But having said that, the level of enthusiasm for this, both in terms of the digital biomarkers and others though, and the interest in pharma in detecting patient populations with therapeutic response implications could not be higher.
I just wanted to add one small point here that's probably pretty well known by everybody, but the construction of pharma is changing considerably from what it used to be, in that there's an evolution towards large pharmaceutical companies being pretty much late-stage clinical development and marketing companies, right? And I think Pfizer in particular is a really, clear example of that. And they're huge, so they're going to have an impact on the rest of the industry. Now, that's not true for every company, and I know Eli Lilly has a long history, especially in CNS drug discovery, and they may be a holdout in terms of moving to this newer model. But the reason I wanted to make that point is because I think it's not necessarily true that you're going to find the best partners in a pharma company. You may actually find the best partners in smaller companies that are invested in a particular platform, or they're invested in a particular disease mechanism. And I would also say that the rising influence of these patient advocacy groups like the Michael J. Fox Foundation or the ALS Association, these are groups that are focused on the disease. And you're right in that the pharma interest will wax and wane on a particular indication based on what the current environment looks like. But if you're Michael J. Fox, you're focused on Parkinson's. You're always going to be focused on Parkinson's. And so I think there's a evolving ecosystem here where the pharmaceutical companies are the pathway to get the new treatment into the hands of patients, but they're not necessarily the start to finish that they used to be. That I just wanted to make that one point.
That's very well said, and I'd agree with that completely.
Susan, from NIH's perspective, I'm curious how you see this partnership working.
Well, let me take a step back, because a lot of what we've heard has been the idea of using technologies to really monitor things once you have a drug, or once you have compounds, or once you have established. But where I see in psychiatric drug discovery, the kind of place where there's a huge gap is getting to that point we don't, getting to the point where you have a compound, you have a target, you've explored things. And so I think one of the things that has come up over the past few years over and over and over is animal models. And I think in some ways we have to change the way we think about animal models, because when I arrived at NIMH, a lot of the clinical researchers there view animal models as a model of disease. And animal models are really models of processes that may be involved in a disease. And so I think one of the things that we would benefit from is to begin to break down models into processes and steps along the way, rather than say, oh, my mouse didn't get ALS. Well, there are a lot of reasons why your mouse isn't going to get ALS. And yet, there are elements that you can test in those models if you design it, that you actually could begin to think about targeting. And there have been some, ALS is one, there have been some recent exploration of antisense oligos. Even though the mouse itself, which is a model of one of the expansion repeat diseases, doesn't actually present with symptoms, you can begin to test whether the interventions are possible. So what I think, I think there's a wonderful and exciting new world of, remote monitoring for testing drugs once you have patients, once you have the kind of the natural history of the disease well worked out, and once you have stratified patient populations, which we heard from Jonathan about the potential for, developing much more stratified views of individual subtypes of the disease. So I'm really looking at it from seeing the failure that we don't understand some of the very basic aspects of the models that we develop, and breaking it down, I think, is the way I would like to see, and encourage and foster.
So you don't think, I gather then, that it's so critical that there's not, for example, a great mouse model that manifests the entire phenotype of a complex psychiatric disease, but if you have animal models that manifest different aspects of processes, you can attack it in parallel that way. Is that?
And really develop a direction and an understanding that will allow you to move things faster into man.
Okay.
And I know Mike highlighted that as well, and that's a strong topic of internal discussion, that these are really not models. In a sense, we've started to call them animal assays, because
Thank
they do
you.
assay a
That's really
particular,
great.
and we're trying to enforce that internally. But I'll give a ray of sunshine there on the remote monitoring. I struggle with how much to say on compounds in development on recorded media at public events. But there is a curious phenomenon to some of the signaling pathways in psychiatry and in dopaminergic circuits, where there's some kind of potentiation that goes on where after the last, so you mentioned maybe every 5 days, there's this phenomenon where after the last dose, you still see the effect. And it can be for several days before it stops. So one of the most impressive things about this biomarker monitoring of the drug effect in this D1 oriented therapeutic that I discussed is when we looked back at some of the preclinical assay, in this case it was a memory test, we found that the effect persisted. And this effect isn't disease, and it isn't necessarily response disease, but it is an effect on drug on pathway, and it persisted for 5 to 7 days. And then when we looked at the data coming off of what was being recorded, these changes in rhythmicity or these changes in gait persisted for 5 to 7 days after treatment, and which meant they left the treatment facility and went back home. You always struggle when you see a change and they've entered a treatment facility, because it may just be compliance. Somebody's giving them drug and watching them take it. But when you see it march out 5 or 6 or 7 days and then go away, and that matches the preclinical assay that you used in drug development, that's a bridge that's very strong. And actually, internally, that's been an extremely strong piece of evidence that's aroused a lot of interest in this area. So I think your point's exactly correct. As you identify better animal assays, there's also an opportunity to bridge those functional assays into human behaviors. And so there's an opportunity to get started early.
Yeah, I know there are a number of people in the audience that want to jump in. I see there's a question in the back. Reed, is that you, I think, up there? Would you stand up, Reed, since you don't have a mic, and boom your voice. Is there a microphone? Yeah, there's one coming towards you. Okay.
For this to work right, the next person to ask a question should be in the front row, and then the next one in the back.
They will be.
Exercise.
It's on. It's on.
Hello?
Yep.
Okay, so first, I thought there's a theme. There was a really interesting theme that ran through all the talks, including the industry talks, which I would like to pick up on and translate into some different words. So in large part, this is a cartoon. In large part, you could talk about the failure of CNS drug development as a failure to recognize the difference between levels of description and the nervous system, number one. Tuning, let me get through it, and you can let you guys respond to it. Tuning a receptor. So you spend a lot of time on understanding the chemistry of proteins and interaction with ligands and intracellular and extracellular signaling cascades, and that is a lush and rigorous area. And we spent 1/1000th of the kind of rigor and attendance and effort in understanding behavior and perception.
Hold the mic closer. Sorry.
Behavior and perception. You need to turn it up. Behavior and perception. So I would offer the following, that there's a big difference between a pathology because you've written a bad software program and a pathology because the memory registers in your computer aren't working right. You could imagine a case where you're acting all kinds of strangely. There's not a single part or signaling cascade in your brain that's dysfunctional. Okay. The reason we don't know how to identify that is we haven't done, and I think like in both the industry talks, we have terrible phenotypes. The reason you can't discover a drug is if you're really in the behavior and perception modification business. Mood, walking, what do I see? When do I panic? When do I feel anxious? How do I perceive threat from others? These are things that live kind of in the psychology world but haven't been given the kind of rigor that has been put on, development of understanding of how molecules and cells compile in. So in large part, I heard yesterday a description of the Kavli Brain Project that's about to launch in New York City. I think they're going to do 20,000 people in the first instance. And this was the issue. We have not differentiated phenotypes well enough yet in order to even understand whether or not we've discovered a drug. And so it's a little bit hit or miss there in that sense. And so I'd just like to get the impression from the industry guys in particular, in your industry, is it not, this is aside from we want to get better biomarkers. A biomarker isn't really a dynamic description of behavior. So instead, why don't we take behavior and perception a little more seriously, literally at the level that we've taken the chemistry and the biology, but because that's what you guys are looking, those are your in?
Well, I'll start and say that the business is asset driven. So there's an incredible focus on what is your product going to look like, right? So that is never going to go away because that's where the reward structure is, what it's based on. So, and in order to identify the best asset, you really want readouts around what that asset is doing in some particular system. So, and it usually you want to have that sort of boiled down to the most minimalistic, scientific parameters that you can line up, and that is, what are the pharmaceutical qualities and, what's its potency at X. But the point that you're making is a very good one. It's just that within the industry, I think it's very difficult to integrate that type of thinking. And in fact, I think where the advances will come first is in a better understanding of the disease. And I intentionally use disease models, not animal models, in my slide for this very reason, that there is a conflation between the two. But we need a better understanding of the basic disease mechanisms, and the biomarkers that I would want to see in a program would be related to those disease mechanisms, not simply just a readout as, did you bump receptor X? So I think the point you're raising is a very good one. It's very complex for an industry that's sort of driven on a focus on the asset. That's my major point.
And I would agree with you as well. Phenotyping is the most difficult thing now in drug development. As you say, you can be moving a phenotype but not measuring it. I presented sort of the traditional biomarkers. I mean, these are phenotypes. You secrete more or less dopamine or DHPC breakdown products. That is a phenotype. It's the result of something, but it's not a behavioral or psychologic phenotype. It's a chemical signature gathered very infrequently in a trial. But the FDA understands it. The test can be validated. And pharma has found itself in this box. The best phenotyping, this future of digital biomarkers is likely based on the model that Google's and other data miners who have, as you've described, a number of people interacting, in this case over the web, and they're tracking a lot of this incidental data, and they're matching it to an outcome, which is likelihood to buy a product, for example, when people invest in these companies, say, we want you to bring us users who are likely to buy our skateboards. And just based on their behavior on the web, Google begins to build associations between if they go to this site, this site, this site, if they register here, if they have this profile, they are more likely to buy your skateboard. It's that kind of reasoning that's very open-ended in the beginning that's likely to paint a fuller phenotypic picture. That's the real hope for these digital devices. And I've presented a very restricted one, but there's no reason that this can't be broadened. There's no, not giving anything away, but there's no reason Lilly can't work with Google. And we can't be one of their customers, and we can't ask things of trial participants along the same lines. What would have to happen, though, to Mike's point, is once you began to paint a phenotypic picture that was predictive, then you would need to get it into something that can pass regulatory scrutiny. And nothing's ever been registered in this category. And so pharma sees this as a tremendous risk if the asset hinges on it.
Okay, thank you for that. But just to follow up, you market it as a behavioral compound. It's called an anti-anxiety drug. It's called an antidepressant. It's not called an anti-5-HT receptor transporter compound. So just to swing the pendulum back a little bit, you already sell these compounds as behavior modification devices, not 5-HT transporter blockers. We just have to know that. So I would offer that you're already marketing it in the domain of this will remove your anxiety, this will move you from being clinically depressed to actually not being able to have a job, this will cure your insomnia or your agoraphobia.
Right, and that's because they can pick an endpoint and a validated clinical measure, and if they hit that endpoint, they get to put that in the label. But I would carry your point a bit further and say, they have these meetings, sometimes they'll look at 2 or 3 indications because they really don't know. What they know is they're inhibiting 5-HT2A. What they don't know is which of these validated measures that allow them to say they're doing this, that, or the other is an appropriate effect. There's incomplete phenotyping. And so they either roll the dice and launch a number of generally underpowered phase 2 studies, which often fail, even if the drug would have succeeded under other circumstances. What they might instead have done is the kind of phenotyping that I think we're both talking about along the core category of drug development, and then from that research rationally pick an indication where you already have some unbiased evidence that this is what your drug's doing. So they often don't have a phenotypic landscape against which to rationally select an indication. They'll do it by market capitalization potential. They'll do it by expertise in clinical trials in this area. A lot of things come into it. The availability of an FDA-validated endpoint. They can say they did that. We can say we've affected Parkinson's disease if we lower the MMSE or the ADAS-Cog sum of boxes by 2 points over 18 months of treatment. What does that even mean? So, patients don't come in and say, I want my ADAS-Cog lowered. They come in and say, I want to remember why I drove to the end of the driveway. We haven't phenotyped that. So there is a third party that has to get involved here, and that's the FDA. A fuller picture of a modern avenue toward indication has to be painted, and the acceptance criteria for the use of these new measures has to be painted. Otherwise, pharma will always be considering this potentially a crucial risk as you move into phase 3. Because it puts a compound at risk that you'll submit it and you do everything right. And the FDA will say, we don't recognize this as a valid demonstration of evidence toward the indication, even though you're showing, no, this is what the patients do when we put them on the drug. they'll say, well, we don't care. We don't understand how you got there. That isn't a validated measure that leads to an efficacy claim. You can't have your drug. So the FDA, and the FDA is aware of this, but they are the slowest moving of the glaciers.
We're going to have to move on to one more question, Tor.
Well, in a way, this is a depressing picture. There's so little new discoveries and such a vast challenge. And I agree that we need better phenotyping. What is the responsibility of other societal institutions and actors, like the policies, the universal leadership, FDA you mentioned? How can we all work in concert to address, for example, the number one cause of disease in depression that's coming up? So how can we move on from where we are now and improve our models, both our disease models, our phenotype models, but also our models of collaboration between government, industry, universities, and individual scientists?
Take a shot at
Take
that.
that one.
It's a challenge.
Yeah, I'm glad you asked that during Nobel Prize week, because the answer to that question probably qualifies.
Yeah, I think it's happening organically already. Again, because there's a shift in the ecosystem and where the investment for researching new medicines is really going, in my view, is towards smaller companies that are funded by venture groups in particular. So these are often a combination of entrepreneurial academics and folks with some level of industry experience that understand what is ultimately going to be a marketable endpoint, and investors that are interested in placing the bets. And when we were having breakfast this morning, we were talking a bit about the fact that the venture groups look at their investments as stock picks, and they really only need 1 of 10 to hit big, and they can have 9 other losses and still do very well. So they're looking for innovations in how new therapies can come out of new perspectives on a particular disease. And once the risk is gone from that, then the pharma industry is extremely interested. And so I only see that ecosystem continuing in that direction, where the big risk is taken by many small players, and the larger pharma are actually there to make sure that product gets to market.
So I can give you the answer that we've come up with, which is essentially to try and move outside of the university. And with a lot of negotiation, I spend a lot of my time now with lawyers, setting up an entity which would essentially function independently of the university, but be able to receive funds from the university, and also, and this is why we're doing it, from venture capital, from drug companies, from whoever wanted to invest it in it. And the aim of that company, risen into the Constitution, is to cure depression. So we think that's one way you could do this. I don't know whether we'll get a Nobel Prize for that.
If you cure
So
depression, you'll be on the short list.
I agree with Mike in particular that a lot of it comes from more nimble biotech and smaller companies. But one thing we've done at NIMH is we've started something we call the Translational Neuropharmacology Task Force. And it's a group of actually advisors from industry. And one of what we do is we solicit from all over sort of innovative new drug targets from either small biotech or can be academic as well. And then we have it vetted by the group. And we also look at our own portfolio and see if it fits in and we can engage our researchers, because we have access to PET and a lot of really terrific facilities. And so far we've had a number of drugs come in. We've started to try to de-risk them. So we can't take them all the way, but what we can do is we can take them far enough that it gives enough data to allow the company to move forward on it or give it a rationale. And so it's this sort of iterative partnership between you know, our institute, and this is just in the intramural program, but it allows us to go back and forth with some of these ideas. And what I like about it is that we get really new targets, ones that I've never, you know, didn't imagine, but they have indications in a number of areas, and if we can work on it, we do. So it fits along with what Mike said.
I think unfortunately, we got a little bit behind time. We're going to have to move on. So please join me in thanking all of our panelists, Dr. Amara and Dr. Miller, Dr. Flint and Dr. Wood. Thank you very much.